and welcome to this lesson for higher biology, which is going to be looking at the two next sections of respiration, the citric acid cycle and the electron transport chain. So we've just finished off looking at glycolysis and the production of pyruvate from glucose molecules. And we're moving on now to look at the citric acid cycle and the electron transport chain. And then later on we'll go back and kind of review all of this as a whole. So what you need to be able to do is describe the citric acid cycle. You need to be able to explain the need for hydrogen acceptors and state where they're used. You need to describe the processes of the electron transport chain, and you need to be able to describe the formation of ATP. So we have glycolysis taking place in the cytoplasm, and the pyruvate itself is then going to be passed on into the mitochondria, and that's where the citric acid cycle and the electron transport chain are going to take place. So when we look at the mitochondria itself, you can see it's got a double membrane. It's got this outer membrane surrounding it, and then it's got this inner membrane which is all folded up. And these folds are referred to as cristae, and they help it provide a larger surface area of what we've got available. So within our inner membrane, around its fold, we've got a matrix, and this matrix is full of fluid which has got the right sort of enzymes that are involved in the citric acid cycle. So the citric acid cycle is really taking place within the matrix of the mitochondria, and we've got on the inner membrane, we have the electron transport chain. So all of the appropriate proteins for the electron transport chain are squashed in and around that membrane. So at the beginning of the citric acid cycle, we initially have our pyruvate molecule, and when we've got oxygen available, it's going to diffuse into the mitochondria. And as it's diffusing into the mitochondria, first of all, we have a carbon and two oxygen molecules being removed, and that's forming our carbon dioxide as waste. And as that's being removed, we're making acetate, which is this one over here. We're also producing some high energy electrons, and our NAD, which we've come across before in glycolysis, is going to capture those electrons, and it's also going to attract some positive hydrogen ions, and that makes a molecule of NADH. So don't worry if you don't understand the chemistry of that, just realise that we've got NAD is coming along and it's picking up some little electrons, and that also helps it attract some hydrogen. So we're forming some NADH. We then have to consider the fact that the acetate molecule, this molecule here, which is being produced from pyruvate, so that we're losing these carbons and oxygens, we've lost hydrogens, and that results in this small molecule here, which is an acetate molecule. Now that's really quite small, and the risk that we have with a small molecule like that is that it might go back out, it might diffuse back out through the mitochondria and into the cytoplasm again. So to avoid that, it attaches itself to a coenzyme, which is this one here, coenzyme A. So we wind up with the group acetyl-CoA. So at the beginning of the citric acid cycle, we have pyruvate diffusing into the mitochondria, it releases carbon dioxide as waste, and NAD comes along and forms NADH. We also have coenzyme A binding to the acetate molecule because it's so small, and that stops it diffusing out of the membrane. So we can then take our acetyl-CoA molecule and proceed into the citric acid cycle itself. So within the citric acid cycle, we have our molecules of acetyl-CoA. And don't worry about the fact that we've got 2 times around here. Okay, this is just the best diagram for you guys in particular, but don't worry about the 2. If you remember, it's really because we've got 2 molecules of pyruvate for every molecule of glucose, and that's really where that's coming from. Okay, so we've got our acetyl coenzyme A, and that's a 2-carbon molecule. It combines with this one here, oxaloacetate. So these two are combining together, and that forms citrate. So we've got 2 carbons and 4 carbons, and that makes our 6-carbon citrate molecule. Now, citrate is going to carry on its way through this series of specific reactions until it forms oxaloacetate again. As we lose carbons from our citrate molecule, we're forming a carbon dioxide molecule, that's our waste, and releasing some hydrogens as well as it goes round the pathway. So as you can probably have guessed by now, those hydrogens are going to come along and they're going to be picked up by our lovely NAD molecule again, forming our NADH. So we've got lots of NADs occurring every time. So the intermediate molecules you don't need to know the names of, you just need to know that at the top we've got citrate, oxaloacetate. Oxaloacetate is combining with acetyl coenzyme A, and that gives us our citrate. Then we lose some carbons as we work our way around, forming oxaloacetate again. We've got 1, 2, 3 intermediate steps, and between each of the lower steps we are losing some of our hydrogen molecules being collected by NAD to form NADH. And that really is just the citric acid cycle. The second part you need to know about is the fact that these hydrogen ions are becoming bound to NAD to form NADH by the use of an enzyme called dehydrogenase. So dehydrogenase is quite an important enzyme in respiration because it's catalysing this reaction, the adding of a hydrogen to our NAD molecule. Now you may also see that hydrogen and electrons might become bound to a different coenzyme. Rather than NAD, you may see FAD, and it's exactly the same. Instead of the N, we've got an F, it's FAD, and it is converted instead to FADH. So from that, we're going through that cycle and we're producing all these NAD and FAD molecules. And we need to get them essentially to this next stage, which is our electron transport chain. That's really, the electron transport chain itself is really just a series of proteins, and they are present in the inner mitochondrial membrane. So all of those NAD and FADH molecules are coming along and passing on the hydrogens and electrons to this set of proteins within the membrane. When we get there, and this is where we get a bit chemistry, we've got our NAD molecules, our NADH molecules, and our FADH molecules. And what happens is the electrons and the hydrogens are basically separated. And the hydrogens go back out across the membrane, and they're essentially going into the intermembrane space. So if we go back to this diagram here, they're getting pushed out, so they're going into this little gap out in here. And when that happens, our electrons are then pushed through all of our different proteins across here. And essentially what that's doing is we're using the energy from the electrons to push the hydrogen sort of against diffusion across the other side of the membrane. Okay, so we want to build up, we want to take all these hydrogen ions and push them out so that they go across here. Now their job when they get across here is to come back again. And when they do that, what they're doing is they're pushing their way through this very special molecule here, which is ATP synthase. And ATP synthase is what is able to take energy and add a phosphate molecule onto ADP, and it forms ATP. Okay, so our ATP synthase molecule is the one which makes ATP. So finally we're getting this promised ATP that we thought we would get when we started respiration. And what the hydrogen molecules are doing is they're pushing their way through this ATP synthase molecule. That makes it rotate, it makes part of it spin, essentially. And when it spins, what it does is it adds this phosphate molecule onto ADP, using up energy, and produces our ATP molecules. And to look at it in a little bit more detail, essentially it looks a little bit like this. We've got lots of accumulation of all of those hydrogen ions that have been pushed back across over here from across all those other proteins, and we've got our ATP synthase molecule, which has got this little rod in it, which makes it spin round. So movement of those hydrogens through this enzyme are going to make it turn round, and it's going to take that phosphate molecule and it's going to produce ATP. Now the last part we need is our oxygen molecule, and our oxygen molecules are quite important, and one of the reasons we need oxygen for respiration to occur is it's our final hydrogen acceptor. So we need this oxygen to come along and take these electrons and these hydrogen molecules away and form water. So without oxygen we can't have that happening. We don't have anything to take away the hydrogen molecules, which means that we essentially wind up with a higher concentration of them on this side than we do on this side. So even though we might get these ones being pushed through, they're not going to come back again because they're not going to go from a low to a high concentration. They need that diffusion action to be able to move through. We also, remember, need our oxygen to help oxidize pyruvate at the beginning of the citric acid cycle. So without oxygen, we've only got glycolysis happening, and that's the only one that produces our 2 ATP molecules. Whereas if we can have oxygen present, then we can get all the way through this pathway, and usually that results in a further 36 ATP molecules approximately. So our total yield in respiration, as you'll know from National 5, is 38 ATP molecules. Now, that doesn't always happen. We occasionally have anaerobic respiration, and that takes place when we don't have oxygen. So it's a little bit different in both plants and animals. So we still have our initial pathway, we've still got glycolysis happening, we've got glucose being broken down into pyruvate. Now in plants, if we don't have any oxygen, ethanol and carbon dioxide are produced. Now the carbon dioxide itself is released by the plant almost immediately, so this process is irreversible. We can't get back to pyruvate and continue down the normal respiration pathway. In animals, however, things are a little bit different. Our pyruvate molecules are converted into lactate, and this reaction is actually reversible. So when we have glucose being converted to pyruvate, and then as an oxygen debt builds up, this forms lactic acid, and however, if we get oxygen back into the cells, they can convert this lactic acid back into pyruvate, and that pyruvate can carry on through the normal respiration pathway. So from that, you should be able to describe the citric acid cycle, the fact that we've got pyruvate diffusing from the cytoplasm into the mitochondria. There we release carbon dioxide as a waste, and our molecule acetate binds with the enzyme to form acetyl-CoA. And that acetyl-CoA molecule is going to combine with oxaloacetate to form citrate. And citrate then goes through a series of reactions to be converted back to oxaloacetate. And as it does, we release some carbon as carbon dioxide, and we also release some hydrogen ions, which are carried by the coenzymes NAD and FAD. Those NAD and FAD molecules are our hydrogen acceptors, and they take our hydrogen and our electrons off to the electron transport chain, where our hydrogens and electrons are separated. Hydrogen ions are pumped across the membrane by proteins using the energy from those electrons. Once they get across the other side of the membrane, though, they just come straight back through the ATP synthase molecule, which is one which spins around as a result of these hydrogen molecules being pushed through it. And it uses that motion to add phosphate onto our ADP, forming ATP. And respiration without oxygen in plant cells gives us ethanol and carbon dioxide, which is an irreversible process, whereas in animal cells it gives us lactic acid, which when our oxygen debt is repaid, can be reversed.